In this video, we're going to do a brief overview of FlyLab JS, which is a virtual simulation of fruit fly genetics. The first thing we need to know about the program is how it designates traits for the flies that you are designing. If you design a fly and you can choose whatever you want in terms of all of these lists of characteristics here, whatever you design is going to be homozygous, meaning it has two alleles for the trait that you are picking. So if you're picking a specific type of bristle, like stubble bristle, the fly that you design, if it says stubble bristle here, that means it is homozygous. It has two stubble bristle alleles. The only exception to that is the lethal alleles. There are some alleles that when a fly inherits two of the same allele, that is lethal. So anything listed in the chromosome map under the lethal trait, like star eyes, for example, that means that when you design a fly with star eyes, it's heterozygous because you can't have a homozygous star-eyed fly that is incompatible with life. Second thing to note is the designation wild type. Wild type just means the normal trait for a fruit fly. Any variation from wild type is a kind of mutation. And so when you're picking all of the different things here, like the purple eyes or the vestigial wings, those are mutations that cause a variation that make it look different from a wild fruit fly. Now, you'll notice these are capital letters. That does not mean it's dominant necessarily. Again, this program is using capital letters for everything because part of what some people do with the program is try to figure out inheritance patterns. So it doesn't want to give it away if the trait is dominant or recessive. So this fly here would be described in this way. If you look at the trait list, notice it's wild type for everything. Those are the normal types of bristles, wild type for the body color. It's wild type for the wing shape, but not the size. Notice the size, it's kind of shriveled away. Those are called vestigial wings. So it says VG for vestigial. And then obviously the eyes are purple instead of red, so it says PR for eye color. This is a chromosome map of the fruit fly. It is at the very end of your assignment. You are going to need to refer to this throughout the assignment because fruit flies only have 4 chromosomes. The first one is the X. So when it says a sex chromosome, that is chromosome number 1 in a fruit fly. And then we have chromosome 2, 3, and 4. All of these traits have been mapped to a specific location on a chromosome. You'll notice these numbers are just basically telling you the distance from the top of the chromosome. You can see as you move down the chromosome, these numbers are getting larger and larger, so you can kind of get a relative distance. Lobe eyes and curved wings, that's really close together, and brown eyes is farther away. Here is the lethal allele list: star eyes, curly wings, dichete, aristopedia, and stubble bristles. So unless you are specifically trying to figure out something with lethal alleles, you want to always avoid anything in this box because you're not going to be starting with a purebred fly. So in the assignment, it's starting you out with a monohybrid cross. This is just your standard 4-box Punnett square. And you want to start with all of the flies just in the normal condition and then go through the steps, follow the steps to actually complete the cross. So we're going to switch over now to that website and see how to perform a cross. So here we are on the website. We have a choice between female and male. So I believe in the first cross we want a female with vestigial wings. So we will go to wing size and find vestigial. And now you can see it has shown up here. And then we're going to select that fly. We want a male with just regular wild type everything. So there we are, wild type everything. We can mate those flies together, and here we see their offspring. So it tells you, it splits them into female and male. Plus means their phenotype is wild type. This is a regular fly. N means number, so 513 of the females are wild type, and then males, 494, our wild type. So we've got our two purebred parents. That's what they look like. They make all hybrid offspring, and all of these offspring are wild type. That tells us something about the dominance pattern, right? If you have a vestigial wing fly and you cross her with a wild type fly and all of the offspring are wild type must be dominant in this case. So what we want to do is we want to select these two flies to mate. These are both the hybrid flies. This is their phenotype, wild type, so they're both a plus, but they're the offspring of cross one, so we know they're heterozygous. We can mate them together, and now we see their offspring. Now we've got 4 different phenotypes here because, again, notice it's separating female and male. If we want to analyze this, we're going to go to the Analyze tab, and it's not sex-linked, so we're going to check this box, Ignore Sex, and now it tells us for wild-type flies there are 779. Here's our proportion, 0.769, and then for vestigial wing flies there's 234, or a 0.231 proportion. So we're going to take this information. You can add it to your lab notes, add comments if you want. That's good if you want to go back and look at it later. But we're going to our Analyze tab, and we now want to check this box for Include a Test of Hypothesis. If we were to do a Punnett square between 2 heterozygous individuals, we know wild type is dominant. We would expect 3 out of 4 to be the dominant phenotype and 1 out of 4 to be recessive. So that is a 3-to-1 ratio. So in our hypothesis, we are hypothesizing from the Punnett square that there are 3 wild type for every 1 vestigial. And then we are going to click to check the hypothesis. It does all the calculations for you, and it gives you a chi-squared value here. 1.95 is our chi-squared value. Tells us we have 1 degree of freedom. So if you go back to your assignment and look at the very end, there is a chi-squared table. We're always using 0.05 as our p-value. 1 degree of freedom gives us a critical value of 3.841. So what that means is our chi-squared of 1.95 is lower than the critical value, meaning that we fail to reject the null hypothesis. We think that our hypothesis of a 3-to-1 ratio is correct. We think that our observed and expected are similar enough to each other because their chi-squared value, which measures the difference between observed and expected, that chi-squared value is very low. It's lower than our critical value, so we fail to reject the null hypothesis, and we think this is the correct explanation of the genetic inheritance pattern. So hopefully this overview was helpful in seeing how to use the program and how to get it to calculate chi-squared for you. If you have any questions as you're completing your FlyLab, please make sure you ask me those questions in class.